Hi and welcome back. This video lecture will be on visual physiology. The learning outcome is to explain how you see, basically how you see color, how you make depth perception, how you get light from striking the retina all the way to processing in the brain. We left off last time with the structure of the lens, and that's where we're going to start. We're going to look at how light changes its path as it travels into and through the eye. The first step is refraction. In refraction, the light rays are bent as they hit the surface of the cornea, as they pass from the cornea through the aqueous humor, as they pass from aqueous humor to the lens, then to the vitreous humor before finally striking the retina. You know how a straw looks when it's sitting in a glass of clear liquid? The straw looks disjointed. That's due to the refraction of light as it passes from one medium, air, into another medium, water, and back out again. The refraction in the eye is controlled by changing the shape of the lens. The lens can become flatter or rounder depending on how far or near an object is as light passes from the object through the eye. The focal point is the point on the retina where the light rays should converge. The focal distance is the distance between the lens and the focal point on the retina. As you look at this image, you're seeing light rays from a distant source. The source is so far away that the light rays are basically parallel as it reaches the eye. The reason is not that all the light rays are parallel, but rather that only the ones that are parallel are going to reach the iris of the eye, pass through the pupil, and hit the lens. The closer the source is, the more the divergent light rays are likely to hit the surface of the lens. So there's a big difference in far vision, which includes anything about 20 feet away, and near vision, which can include some of those divergent light rays. The focal point, remember, is on the retina. So accommodation is the change in the shape of the lens that allows the eye to focus on an object whether it's near or far. In astigmatism, the surface of usually the cornea, but sometimes the lens, is irregular, and the light rays don't all run straight because of the irregularities of the surface. This distorts the image. Astigmatism can be treated either by specialized glasses that correct specifically for an individual's astigmatism, or these days by changing the shape of the cornea with laser surgery. Now, for close vision, you're going to see that the lens is quite rounded. For far vision, the lens is flattened. So flat for far and round for near. As you can see in close vision, the rounding of the lens allows for greater refraction because the light rays, as they come in, are more divergent. They spread out further, so they all have to be brought back together to hit the focal point. In order to do that, the ciliary muscles contract. With the light rays coming in parallel from a far object, there's less rounding required. So the ciliary muscles relax, and that allows the flatter lens to simply pass the light rays through to the focal point. For this reason, near vision requires contraction of the ciliary muscle. Far vision requires relaxing the ciliary muscles. Near vision is associated more with eye strain, and you're encouraged, if you've been reading for a long period of time, to focus on something in the distance to let those little muscles relax. So if a far object is 20 feet away, then that becomes the point at which we measure whether your eyes are normal compared to other people's. So if your vision is 20/20, then you can see at 20 feet what other people can see at 20 feet, as judged usually from an eye chart. If your vision is better than average, you may have 20/15 vision, so you can see at 20 feet what other people have to be 15 feet, 5 feet closer to see. If your vision is worse, you may have 20/100 vision, where you have to get 20 feet to see something that other people can see from 100 feet away. Because the lens is the sort of the hinge of the focal plane, objects at the top of the visual field are going to be focused on the bottom of the retina, and vice versa. So images are reversed as the light strikes the retina. And of course left and right are backwards as well. In myopia, which is nearsightedness, The eyeball is too long, and the focal point of the normal lens is trying to hit where the retina should be, but the retina is further back. A concave lens will correct for this condition. In hyperopia, farsightedness, the eyeball is too short, so the focal point is trying to hit a retina that is in fact too close. To correct for this, we use a convex lens. Moving on to what happens when light strikes the retina, assuming it strikes it in focus. Remember those rods and cones, the photoreceptors of the retina? Rods can see in dim light. It doesn't take a lot of energy to stimulate a rod. However, cones need more energy. Furthermore, there are 3 different kinds of cones, each with its own range of sensitivity, so that you can see different colors, different wavelengths of light. Whether it's a rod or a cone, within the cell there is a collection of the visual pigment. Visual pigment is a photosensitive molecule that changes its shape when struck by photons of light. In the rods, the visual pigment is called rhodopsin, and rhodopsin is synthesized from vitamin A. This is why vitamin A is considered essential for night vision. The cones have each their own visual pigment, slightly different from rhodopsin, each sensitive to a different wavelength of light. In this image, you can see a cone and some rods. Their tips are embedded in the pigmented epithelium, that pigment layer of the retina. Remember that light is coming through from bottom to top in this view. Each rod and cone has an outer and inner segment. Now, this is too much information for test purposes. Basically, when the light strikes, if it's dim light, it will hit the rods. If it's bright enough light in a certain wavelength, it will hit the cones and excite these cells and create an electrical change, which can then be picked up by the bipolar cells. So back to those cones again. The 3 wavelengths that can be detected by cones in humans are red, green, and blue. So red wavelengths, blue wavelengths, and green wavelengths can strike different cones, and as they do, they send impulses to the brain that are interpreted as color. Color blindness occurs when the cones of one of those 3 kinds is either missing or mutated. The most common example of this is red-green colorblind, in which either the red cones or the green cones have mutated so that the color interpreted is for the red cones is green or for the green cones is red. So whether red light or green light, it all looks the same to the brain of someone with red-green colorblind. Here you can see the wavelengths of light. We're not really going to worry about this for the test, but just so you can say you saw it. The wavelengths of light range, visible light range from about 400 to 700. The longer wavelengths are in the red end of the spectrum. The shorter wavelengths are in the blue end of the spectrum. So blue cones, green cones, and red cones each have their own preferred wavelength. Rods have pretty wide wavelength sensitivity and can in fact go all the way to not quite the red end, notice. So the next time you're in the dark, notice that the reds vanish pretty quickly. Here's a typical visual test. If your vision is normal, you should be able to see the red dots forming the number 12 amongst a gray background. But imagine that that gray background was in fact different shades of green. Someone with red-green color blindness would not be able to see the numbers 1 and 2 in the field. Now, once light has struck the rods and cones, there's a recovery period. And we say that the color's kind of bleached out. It takes a moment for the molecules, the rhodopsin molecules, to recover. And during that time, then you're kind of dazzled by light. So you might notice that if you're in the dark and someone turns a bright light on, that momentarily you can't see. And then if the light goes off, it takes a while to get accommodated to the dark again. That's the bleaching from the recovery period. In people, particularly older people, but definitely people with deficiency of vitamin A, night blindness is more common. It just takes so long for the rods to accommodate that really people can't see well at night at all. Many people have trouble driving at night because of this. If the headlines shine in your eyes and it takes too long to recover, then you can't see the road very well. In adapting to light and dark, part of the adaptation is change in the size of the pupil. Remember, this is accomplished by the smooth muscle of the iris in two layers, the radial muscles and the circular muscles. Now, let's look at the visual pathway. It starts, of course, at the retina. As light strikes the rods and cones, it ends in the brain. And the most sophisticated terminus in the brain, of course, would be the cerebral cortex. The visual cortex in the brain is in the occipital lobe of both the right and left cerebral hemispheres. There's a primary visual cortex. There are secondary visual cortical areas for processing visual images. Our understanding of visual processing is pretty sophisticated now and much deeper than what we need to know for the test. So if you're aware that the terminus is the visual cortex and the occipital lobes of the cerebral hemispheres, you're in pretty good shape. And we need to look at the pathways in case anything were to happen to interrupt that process. So the first step is from the photoreceptor, remember that's the rods and cones, to the bipolar cells, and from the bipolar cell to the ganglion cell. All of that is still in the retina. The ganglion cells then, their axons converge to form the optic nerve. They go back through the back of the eye. Optic nerve is cranial nerve 2. It proceeds from the posterior surface of the eye to a point called the optic chiasm where they cross each other. That's going to occur at the diencephalon. From the optic chiasm, optic tracts will pass to the thalamus. From the thalamus, the optic radiation will carry the impulses to the occipital lobe, right and left visual cortex. Depth perception is achieved by comparing the right and left sides. Here in the picture you can see the visual fields. Let's take a look. The left eye only sees the far left peripheral part of the left visual field. Only the right eye can see the far right side of the visual field. The combined visual field is the area of binocular vision where depth perception occurs. Now I want you to follow the little dotted lines. If you see something in your left visual field, then it's going to strike the medial side of the left eye and it's going to strike the lateral side of the right eye. And the same for the other side. On the right visual field, impulses will strike the medial side of the right eye and the lateral side of the left eye. So we want to follow the medial of one eye with the lateral of the other eye, and let's follow those pathways and see what happens. The medial side of the left eye crosses at the optic chiasm. The lateral side of the right eye does not cross. So whether you saw it with the medial side of the left eye or the lateral side of the right eye, those pathways go to the right side of the brain. So anything you saw in the left visual field winds up in the left side of the brain, and likewise anything you see in the right visual field ends up in the left side of the brain. Here are your optic nerves. Here's the optic chiasm. Here are the optic tracts. These little blobs here, the lateral geniculate nucleus, is part of the thalamus. This term, lateral geniculate nucleus, is not on your test. The thalamus is. So in the thalamus, what we're going to see is that the processes take a couple of or three different routes. First of all, they send offshoots to the diencephalon and brainstem, and this allows you to respond subconsciously to the long days of summer or the short days of winter. So it allows for seasonal variation in things like your melatonin production on the pineal gland. Secondly, visual impulses go to the superior colliculi. These, you remember, are part of the midbrain, and they're responsible for visual reflexes. One thing they do is they help you track moving objects, so you do that subconsciously. The conscious impulses go through that optic radiation, and this is where most of the pathway is going to be. The optic radiation goes to the left and right cerebral hemispheres, to that visual cortex in the occipital lobe. So, as I said, some of the impulses do go to the deeper parts of the brain, the more primitive parts of the brain, such as the pineal gland, which is in part responsible for circadian rhythm by secreting melatonin. The hypothalamus also has some kind of biological clock in it, and these clocks get reset every day when the sun comes up and every night when the sun goes down. So you stay on a pretty much 24-hour cycle because you're constantly resetting that clock every day. Next up, the ear.